Okay, today we're going to talk about the tools of geneticists. So yesterday we talked about genetic disorders, and so these are the tools that are used to help out with genetic disorders and other types of genetics technology. So the first thing I want to talk about is pedigree charts. So a pedigree chart is a visual that allows you to track a trait through generations of a family. And on a chart like this, the males are always in squares and the females are always drawn. The symbol is a circle. And if it's a completely filled in square or circle, that means the person has that trait. If it's a half colored circle or square, it means that they are carriers. They're not expressing the trait. It's not in their phenotype, but they have at least one recessive allele, probably hidden by the dominant allele. If they're empty circles or empty squares, it means they do not exhibit the trait. They're not expressing it, and they are also not carriers of that genetic disorder. So here's another example of what a pedigree chart looks like. And again, you see the circles and squares, the half shades, the full shades, and no shades, as we discussed. But this graphic does a nice job of explaining the fact that these horizontal lines between a circle and a square indicate some kind of a union, right? Maybe a marriage or a union of sorts that produces offspring, indicated by the vertical line, and then they're connected by that vertical line. So the siblings are then connected here, 1, 2, and 3. Okay, and then you have these other circles and squares that kind of come in here, and they are people that marry in or further unions that produce other offspring. Not necessarily part of this line, but from another line that marries in or gets together and produces offspring with these other siblings. Okay? So a pedigree chart like these are really useful when you want to talk about something that a genetic disorder that can be kind of debilitating, something like hemophilia, where you don't have the ability to clot or you clot very slowly. So if you had hemophilia in your family line, you might want to consider, what are the chances of producing a child that has hemophilia or producing a child that has cystic fibrosis, because these can be very, very debilitating throughout that child's life. So, depending on your family history, the person that you're going to marry or get together with, their family history, you might want to know these things before you have children. So pedigree is useful for geneticists. You can understand how a trait's inherited and predict the probability of possibly having an organism and offspring with that trait. Pedigree charts are also useful in breeding of plants and animals. So if you're trying to use pedigrees, you want to do it to increase yield and nutritional content, things like that. So if you look at this graphic of this dairy farm with these cows here, if you're a dairy farmer, you want to produce cows that produce a lot of milk and produce good tasting milk or delicious milk and maybe even nutritious milk. So you would want to, if you looked at your cows producing milk, you'd say, hey, wow, look at the milk this cow produces, and make sure that you mated it. And you would mate that cow with another cow that also produces a lot of delicious milk. And if you mate them together, the hope is that the genes will mesh and you'll produce further cows that have that delicious milk. So that's kind of how pedigree charts are used. And when we decide which organisms we want to breed together, we call that selective breeding. Some other examples of that: if you have a pedigree dog at home, if you have a Chihuahua or a German Shepherd or a Collie or a Labrador Retriever, any of those kinds of dogs, and it's purebred, well, it probably has pedigree papers. And that just means that that dog comes from a long line of dogs in that pedigree and has not been crossed or contaminated with any other genes outside of those pedigrees. So thoroughbred racehorses is another example. So racehorses that run in the Kentucky Derby and other Triple Crown races, those are your utmost champions. They come from pure bloodlines. They are only mated with other champions usually, and the hope is that you always get super fast horses.
The Kentucky Derby horse sounds like put in the morning one. Like it was supposed to be like last minute and you're supposed to get like last.
Right. So just because you have a champion bloodline doesn't guarantee that you're gonna, be the winner, right? So, certainly other horses can do that. And that's kind of the thing with pedigrees and with selective breeding is you don't always know who's going to be the best horse or produce the best cow for producing milk or the best cattle beef. Sometimes this can happen outside of those bloodlines, right? So another tool of geneticists is genetic engineering, where you take the DNA or genes from one organism and place it into another. Probably the most famous example of that is by making insulin. So insulin, as you may know, is your pancreas makes it, and it's responsible for controlling your blood sugar levels. And people that have diabetes don't produce any insulin or don't produce enough insulin and can't control their sugar well. So they have to take insulin pills or insulin shots. So that insulin used to come from pigs. They used to take the pigs from, I guess, leftover from when they slaughtered them for bacon and ham and whatnot. They would take the pancreas and they would kind of extract whatever insulin was in the pig pancreas, and that would become our insulin medicine. Today they don't do that. Today they use bacteria, and this is how they do it. Bacteria have their own DNA, but they also have these circular pieces called plasmids. So they use what's called the restriction enzyme, a specialized protein. In this case, the restriction enzyme cuts the plasmid in a particular location. Well, if they've isolated the insulin gene in humans, and they can give it that same kind of complementary stranded end, they call those sticky ends. And if they put that insulin inside here, it'll stick right into the plasmid. And so this is showing a picture of a genetically engineered bacteria containing an insulin gene inside of its plasmid. And so what's going to happen is the bacteria will go about its business making its own proteins as it reads this red circular part, carrying out its life functions. But when it gets to the yellow, it's not going to know any different, and it's going to make insulin, human insulin. And so if one bacteria cell does this, not a big deal. But when you have lots of bacteria cells doing this, you can make quite a lot. And bacteria reproduce asexually, so you get genetically identical bacteria, all containing the insulin gene. And they also reproduce quickly, so you can get millions and trillions of bacteria in just a couple of hours producing as much insulin as you need. And that's why today insulin is very cheap and very available for anyone that needs it. Another up-and-coming genetic technology is new. It's called Crispr, and CRISPR stands for Clustered Regularly Interspaced Short Palindromic Repeats. And I know that's a mouthful, and I'm not going to get into the whole technology for you, but I do want to point out this one word in here, palindromic. And so palindromic means palindrome. Anyone know what a palindrome is? Palindrome is when a word that reads forward and backward the same way. So, for instance, the word race car. If you read it forward or read it backward, it spells race car. Or mom, or dad. Those are all palindromes. Pop is a palindrome. All right, and there are many, many others. So these palindromic sequences are these sequences of DNA that read the same forward and backward in bacteria. And scientists have been able to use those to mark DNA in humans and basically use it to cut out sections of DNA and insert sections that they want. So you can imagine if you had a genetic disorder, and they're not quite doing this yet, they are doing this on some level, but imagine if you had sickle cell anemia and your cells are sickle shaped, that crescent moon shape, instead of the nice oval shape that red blood cells should be. Well, if we could cut out that section of your DNA that codes for making the sickle cell, and we can put in a corrected gene, we can cure the disease. Same thing for cystic fibrosis. If you can cut out the segment that says produce thick mucus in your lungs and substitute that for the normal thin mucus genes that'll code for that, you can cure cystic fibrosis. So they are currently using CRISPR to also help with some types of cancer, where they come in and they cut out the broken part of the gene in the cells that are becoming cancerous, and they put in corrected DNA information so the cell starts behaving normally. So, this is going to be really in the next 10 years, you're going to hear a lot about this, and you're going to see a lot of applications for helping people with genetic disorders. And another thing I want to talk about, another tool is cloning. So we talked about selective breeding, how you can go ahead and take two animals and breed them and hope the best for the offspring that you're going to get the traits you want. Well, in cloning, you don't have to hope. If you can copy the genetic material of a single animal that you like, that has certain traits, you can go ahead and get exactly what you're looking for in the new organism. So what happens in cloning is, and the first animals they did this with were sheep, and that's why this sheep is named Dolly, because Dolly was the name of the first cloned sheep. About 10, 15 years ago, they first did this. But basically what they did was they take a they remove an unfertilized egg. They remove the DNA from that egg. They then take the DNA from the mitochondria of the sheep they want to clone, and they fuse these together, basically fusing the DNA into those egg cells. They then take that fertilized egg cell and they culture it. Well, it's not really fertilized, but it's a clone. So they put it in culture and they make the clone grow, like after a zygote fertilized egg and sperm, that would be one cell, but then the cell would start to quickly divide, and that would form what's called a blastocyst. And the blastocyst is basically just a group of cells that are undifferentiated. You don't have bone or muscle or eyes or any kind of differentiation. They're just cells that are ready to divide. And they put that into a surrogate, a sheep that's going to then carry the fetus to birth, and then you get the new sheep, and the new sheep named Dolly in this case has the genetic material of sheep X, but use the egg and the surrogate mother of these two sheep, but it's identical to this sheep X right here. Yeah. What? Oh, can you do it with humans? Yeah, so that's a good question. So a couple of people have been asking this all day, and so the quick answer is no, because it's illegal. You're not allowed to clone humans. So it's kind of been outlawed that you even work on it. That being said, I can't imagine that in some country, in some remote laboratory, in secret, that they're not trying to do this. But if they did do it and the technology got out, there's a lot of ethical implications. So I'm glad you brought this up. Consider the fact that, first of all, who gets to be cloned? Does anyone who's sick and wants to live longer get to be cloned? Well, now we have a population problem, right? Or do only the rich get to be cloned because they can afford it? Is that fair that you can't afford it, right? And then why are we cloning? Because we want to have more people like us? Well, will they really be like us? Twins aren't necessarily the same. There's lots of influences, worldly influences that influence your personality and your looks and everything else once you're outside the womb, right? And then the last thing is, well, some people say, well, in case I have a heart attack one day or need a liver transplant, I want to have a clone ready so I can have those organs. But that's now a person, right? So do they now have rights as a person, or do they not have rights because you're the reason that they're alive in the first place, right? Because you asked them. So there's movies made about this stuff for good reason. It's really ethically, it's very difficult, right? And creepy.
I feel like cloning just wouldn't work.
It could, it could work. It definitely can work.
Doesn't make sense.
Yeah. So just to recap real quick, selective breeding is when you take organisms with desirable traits and you breed them together in hopes that you're going to get offspring with those desirable traits. Cloning an organism is more specific in that you get the exact organism with the traits that you want. Whereas genetic engineering is when you borrow genes or DNA from one organism and place it into another. And there's big hope for this for curing lots of diseases in people. And the last thing I want you to know is something we haven't talked about yet, but how did we get all of this knowledge? How are we doing all this stuff? About 20 years ago, we had a project called the Human Genome Project. I'm sorry? Dolly was a little bit later, but first had to come the Human Genome Project, where they basically had to map out all the genes in human beings, the whole genetic code. And that was a huge undertaking, and they didn't think they'd be able to do it, and I think they wound up doing it even faster than they thought. But anyway, that's kind of the precursor to all of this stuff that's going forward with genetics and genetic engineering. So, any questions?